Rana Liaquat Ali Khan, Government College of Home Economics, the most reputable and prominent college of Sindh, is the institute from where I belongs to. I, Sadia Idrees, Assistant Professor, Nutrition and Dietetics Department, presenting the topic Protein for BS 5th semester, the course Advanced Nutrition 1. This is common basic pattern of amino acid. This baseline pattern build around a central alpha carbon. At one end of alpha carbon there is amine group NH2. On the other end there is a carboxyl group COOH. All amino acids contain the same common pattern except R. R is a symbol of radical, a variable group. Each amino acid has a different variable group, which make them entirely different from each other. For example, glycine carry only one hydrogen atom at this place, whereas alanine has only a methyl group CH3 at this place. Here there is a list of essential and non-essential amino acids in humans. Essential amino acids are those which are needed to take in the diet. That's why they should call dietary essential amino acids. Non-essential amino acids are also important for the body. But body can manufacture and make non-essential amino acids by itself according to the demand of the body. Leucine, isoleucine, valine are examples of branch side chain and called branch chain amino acid. These amino acids are used in special amino acids product for treatment of liver disease or kidney failure. They also reduce the muscle protein breakdown and speed recovery of the hospitalized patient stressed by severe illness and injury. Threonine and lysine. Threonine has an attached hydroxyl group just as water does. Lysine has an extra nitrogen atom at the end of long carbon chain and it is precursor of amino acid carnitine. Phenylalanine and methionine. Phenylalanine is involved in genetic disease of newborn phenylketonuria. It is a precursor of non-essential amino acid tyrosine. With the tyrosine, it forms the hormone thyroxine and epinephrine. Methionine is a major source of dietary sulfur. It is involved in sulfur containing renal calculus cystine stone. Is genetically predisposed person. With the lysine methionine synthesize carnitine, a non-essential amino acid. Tryptophan is essential amino acid has double ring side chain. Tryptophan is also unique in its capacity to produce the B vitamin niacin as needed. It is also precursor of vasoconstrictor and neurotransmitter serotonin. Serotonin is a chemical that has a wide variety of function. One of them is that it contributes to well-being and happiness. People who take tryptophan as a natural antidepressant, if they take it in excess amount, it affects the neuromuscular problems and even cause death. Histidine is an essential amino acid. It has a ring in its side chain and provides more nitrogen. It stabilizes the attachment of iron in the hem pocket in the formation of hemoglobin. The dual chemical nature of amino acid means an amino group at one end and carboxyl group at another end enables amino acid to join in the chain structure. So, The amino group of one amino acid join the carboxyl group of another amino acid. This characteristic of joining made bond called peptide bond. Here you can see when one amino acid joined to another amino acid through a condensation reaction that gives off water molecules and form peptide bond. When only two amino acid join, the bond is called dipeptide. When one more amino acid linked, it formed tripeptide. And when long chain of amino group linked in this manner are called polypeptide. These polypeptide chains vary in size. 3 to 10 amino acid containing chains are called oligopeptide. Medium sized polypeptides such as insulin, which has two chains of 21 and 30 amino acids respectively. Large complex protein consists of several hundred amino acids. The primary structure is the basic sequence or order of amino acid in their polypeptide chain, formed by initial linkage of the specific amino acids in their specific position in the chain. This process governed by the cell nucleus material deoxyribonucleic acid DNA and its companion ribonucleic acid RNA, which control genetic design. The secondary structure is the folding of polypeptide chain into coiled or plated arrangement, linked to one another by cross bonds, usually of hydrogen or sulfur. Here there is a picture of alpha helix and beta plated sheets. The overall three-dimensional structure of polypeptide is called tertiary structure. The tertiary structure is primarily due to interaction between the R group of amino acid that make up the protein. These include hydrophobic interaction, ionic bonds, hydrogen bonds, and disulfide bridge formation. The quaternary structure of protein is association of several protein chains or subunits into a closely packed arrangement. Each of the subunit has its own primary, secondary, and tertiary structure. The subunits are held together by hydrogen bond and the forces between non-polar side chains. Protein classification according to the function. Number one is a structural protein, collagen connective tissues, contractile protein, myosin present in the muscle, antibodies, gamma globulin which made immune system, blood protein, albumin, fibrogen, hemoglobin, and hormone like insulin, thyroxine, gastrointestinal peptidase work as a regulator. Enzymes, all enzymes of digestion and cell metabolism. Biological value provides a measurement of how efficiently the body utilizes protein consumed in the diet. A food with a high biological value correlates to a high supply of essential amino acids. The biological value also measures a protein maximal potential quality. The high biological value proteins are fish, chicken, meat, egg, milk, etc. On the other hand, the low biological value protein doesn't provide all essential amino acids. So, the vegetables and fruits are the examples of low biological value protein. Classification of protein based on shape and structure. The helical or pleated shapes like myosin in the muscle and globular or round shape like hemoglobin, hormones, enzymes. Classification of protein based on chemical composition. Basically, there are two classes, simple protein and complex protein. Simple protein contain only amino acid and their derivatives like serum albumin, insulin and the enzymes. Complex protein are made up of simple protein plus some other non-protein groups. Examples are nucleoprotein, where the protein combine with the nucleic acid such as DNA, glycoprotein and mucoprotein, where the protein join with the carbohydrate such as mucin found in the secretion of mucous membrane. Phosphoprotein, protein and the phosphorus join together, example is casein in the milk. Chromoprotein, when protein and a chromic or pigmented group join, for example, hemoglobin in red blood cells. Lipoprotein, protein with the triglyceride or other lipid-like substances, for example, phospholipid and cholesterol. Metalloprotein, protein with the metals, minerals such as copper, zinc or iron. Example is Heme, iron binding protein of hemoglobin. Dear students, the metabolism of protein, the dietary protein requirement, and the vegetarian diet, these topics we will cover later on in our upcoming presentation. Till then, Allah Hafiz, Jazakumullahu khaira.